Welcome back, everyone. Today, we're taking a deep dive into a fascinating case study. We're talking about a recently described ocular neurological syndrome. It's been found in a certain breed of dog.
And this is really intriguing because
Yeah.
it has implications for how we understand human neurological disorders.
It does. So we're kind of looking at, like a naturally occurring model
Yeah, exactly. Yeah, a naturally occurring model
that we can kind of use
that can teach us about,
to be like.
yeah, the interplay of genetics, metabolism, and neurological function.
Precisely.
Yeah.
And to guide us through all of this,
Yeah.
we've got, a leading expert in canine genetics and neurology here today.
I'm happy to be here. This case is it's a very interesting one. It really does warrant a closer look, so.
So let's jump right in and meet our patients.
Okay.
Right.
Yeah.
We have two Cirneco dell'Etna dogs, which is a breed I had not heard of.
Right. A lot of people haven't. It's a very rare breed.
It is, which makes this even more interesting, right?
Yes, for sure.
Right.
It's a Sicilian breed.
Let me see.
But both of these patients were males and from the same litter. And the interesting thing is they both began to exhibit signs of vision deterioration around the age of 4. But even before that, they were experiencing these odd neurological episodes.
So the neurological episodes started first.
Yeah, much earlier, around like 2.5 years of age. And they weren't your typical seizures, so.
Okay, so tell us about that. What was going on with those episodes? What were people seeing? The owners, the vets.
So they were characterized by head tremors and involuntary limb movements, almost like sudden, intense cramping,
Oh, I see.
But the thing is, the dogs remained fully conscious during these episodes.
Oh, wow.
And so the vets debated whether these were atypical seizures or a form of paroxysmal dyskinesia.
Okay, so paroxysmal dyskinesia? So that's kind of a sudden involuntary movement disorder, right?
Right, exactly.
Wow. So dramatic. It sounds pretty dramatic. How long does those episodes last?
Not very long, actually, only a few minutes.
Oh, okay.
Which,
brief.
Yeah, they were brief. And what's even more striking is the dogs would recover quickly. So this transient nature of the episodes, it adds another layer of complexity to the case.
Yeah, I was going to say, so now we've got vision problems at 4.
Yeah.
Neurological problems.
Yeah, much earlier.
Starting earlier and not your typical seizure.
Yeah.
And brief.
Right.
And then they're fine.
Yeah, exactly.
Okay, so now let's bring in the family. Let's bring in the family history. The parents of these dogs were not affected.
No.
But they shared a grandmother.
They did. That is a key detail. This pedigree information really suggests an autosomal recessive.
Inheritance pattern.
Yes, inheritance pattern.
Okay, meaning that both parents were carriers of a mutated gene without exhibiting symptoms.
Right. So they were
silent carriers.
Silent carriers. And unfortunately, both affected pups inherited 2 copies of this mutation.
Oh, wow.
Yeah.
Okay, so both parents silent carriers. Now, were there other inheritance patterns?
Oh, yeah.
That they looked at, that they considered?
We considered other possibilities. We thought about, autosomal dominant X-linked recessive inheritance.
Okay.
But based on the family history, the fact that both affected dogs were male, those possibilities seem
less likely.
Less likely, yeah.
Okay. So the investigators, they had a pretty strong suspicion that this was genetic.
Yes.
But how did they find the genetic culprit? Especially in a rare breed like the Cirneco dell'Etna.
That's the question. It's like trying to find a needle in a haystack.
Yeah.
But with modern genetic tools, we can kind of narrow down the search.
Yeah.
So they started with homozygosity mapping. And essentially, they were looking for shared stretches of DNA in the affected dogs that were absent in the unaffected dogs. Yeah. And this pointed them towards specific regions in the genome where the mutation might be lurking.
It's like a genetic map.
Exactly. Yeah, it's like using a map.
To find the treasure.
To find the treasure, exactly. However, working with a rare breed does present some challenges.
Yeah, because I would imagine that it would limit your sample size.
Exactly. The limited sample size makes it tricky to pinpoint the exact mutation.
Okay.
Yeah.
But they persisted, right?
They did.
They did. And ultimately, they found something.
Yes.
They identified a single base pair deletion. And it was in this gene called the AMPD2 gene.
Yeah.
And they found it in the affected dogs. So tell us about that.
Yeah, it was a pretty big breakthrough because this single base pair deletion, even though it's small, it can cause something called a frameshift mutation. You know, it's like imagine you're reading a sentence and one letter is deleted.
Right.
The whole meaning changes, right?
Totally, yeah, yeah.
Similarly, this deletion scrambles the genetic code, leading to a truncated, non-functional protein.
Oh, wow.
Yeah.
Okay, so that's really messing things up at the very, like, basic level of you know, the DNA.
Yeah, it's disrupting the most fundamental level of the code.
Wow.
Yeah.
And so this specific deletion, it was found
consistently in the affected dogs.
In the affected dogs.
Yeah.
So then that makes it the prime suspect.
Exactly. The fact that all the affected dogs had the mutation and none of the healthy dogs did really supports a direct link between the AMPD2 variant and the syndrome.
Yeah.
Yeah.
Okay. So this AMPD2 gene, what is this thing? What does it do? And how is it connected to...
So the AMPD2 gene, it provides a blueprint for an enzyme called adenosine monophosphate deaminase 2. And this enzyme is involved in the pathway that produces ATP and GTP. And
those are like the energy currency of the cell.
Exactly. Yeah. They're the primary energy sources for cells.
So it's involved in energy. That sounds pretty important.
It is. When this pathway is disrupted, it can have cascading effects.
Oh.
Particularly...
So like a power outage.
Yeah, exactly. In tissues that have a high energy demand.
Okay.
So like the brain and the retina.
Okay.
So this could potentially explain the neurological symptoms and the retinal degeneration we're seeing in these dogs.
Makes sense.
Yeah.
Okay, so this one little base pair
tiny
deletion can really throw a wrench.
It can really throw a wrench in the cellular power grid.
Wow.
Yeah.
Okay. And then there's this this link to pontocerebellar hypoplasia in humans, or PCH.
Right. PCH.
Can you talk a little bit more about the similarities and differences between PCH and what we're seeing in these dogs?
So there are some interesting parallels, you know, because in both cases, we see these neurological symptoms that point to...
disrupted brain function.
Yeah, disrupted brain function.
Yeah.
But there are some notable differences. So, for example, the retinal degeneration in these dogs, that's not really seen in human PCH cases.
Okay. So it's not a common finding.
Right.
Okay.
And it underscores the complexity of genetics and the variability in how diseases manifest, even across...
different species.
Yeah, different species. Okay.
So now let's dive deeper, right? Or we've talked about the patients generally, but let's get into some of the clinical details.
Okay.
Can you walk us through each dog? What were the neurological findings?
Sure. So let's start with the first dog.
Okay.
CRN4. He started having those odd episodes a little after 2 years of age, and the owners described a variety of signs, including head bobbing, vocalization, and even lifting of the right front limb as if he was having cramping.
Oh, okay.
Yeah.
And did those progress?
They did. Eventually, the episodes affected the left front limb as well, and the episodes themselves would occur in clusters over a couple of days, lasting for a few minutes each.
Right.
Then they'd subside and then reappear weeks later.
Wow. So it wasn't a very predictable...
Yeah, it was unpredictable and quite distressing, I would imagine, for the owners.
Okay. So then when CRN4 had his neurological exam, what did that show?
So that was interesting because the neurological exam was mostly unremarkable.
Oh, okay. So everything looked pretty.
Yeah. Mental state and cranial nerve examinations were normal. Gait and posture were also normal.
Oh, wow.
And the only consistent finding was cervical spinal pain.
Okay.
So based on those findings, you know, the preliminary diagnosis was atypical seizure activity.
Okay.
Or
paroxysmal dyskinesia.
Paroxysmal dyskinesia.
Okay. So sort of like mixed findings.
Yeah, mixed bag, you could say.
Yeah, yeah. And then... so did they do any other tests?
They did.
Like an MRI or a spinal tap?
Yeah, they did an MRI of the brain and cervical spinal cord, along with a cerebrospinal fluid analysis, a CSF analysis.
Okay.
Yeah.
And what did those show?
So the MRI showed a small area of bilaterally symmetric T2 hyperintensity in the periventricular white matter tracts.
Okay.
And this signal abnormality, extended toward the internal capsule, where it was accompanied by increased T2 signal within the caudate nuclei, bilaterally, more prominently on the right side.
Okay, so there was something abnormal.
Yes, definitely some abnormalities were detected.
Yeah.
There was also moderate ventriculomegaly.
And so...
which is just the enlargement.
Enlarged ventricles.
Yeah.
Right. In the brain.
In the brain, right. And this appeared to be caused by a diffuse thinning of the cerebral white matter.
Okay.
Particularly noticeable in the ventral temporal lobe.
Okay.
Yeah. And
what about the spinal tap?
So the CSF analysis, all of the results were within the normal range, and the MRI of the cervical spine didn't show
anything abnormal.
Any abnormalities? No.
Okay, so some interesting MRI findings.
Yeah, some intriguing findings, but
not crystal clear.
Yeah, not clear answers yet.
Okay, and now let's move on to dog number 2.
Okay.
CRN6. So when did his neurological signs start?
So CRN6's early signs started around 2.5 years of age, and he was examined by a neurologist at 4 years old because of concerns about visual impairment and sudden atrophy in the left temporalis muscle.
Okay, so the left temporalis muscle, so that's one of the chewing muscles, right?
Exactly, yeah.
Okay. And then I also had here that they thought that maybe he had left facial nerve paralysis.
Yeah, so that was later attributed to a traumatic incident where he fell into a cellar.
Oh, no.
Yeah.
Okay.
So it was unrelated.
Unrelated. Okay. So then what was going on neurologically?
When the neurologist examined CRN6, they noted normal mentation, but they confirmed that left-sided atrophy of the masticatory muscles, those are the muscles for chewing.
Okay.
And he also had incomplete pupillary light reflexes, and similar to CRN4, cervical pain.
Oh.
Yeah. And then his gait analysis revealed an upper motor neuron gait affecting all 4 limbs.
Oh.
And it was more pronounced in the thoracic limbs
than the pelvic limbs.
Okay.
He also exhibited crossed extensor reflexes in all 4 limbs.
Okay.
Which is a reflex that often...
an upper motor neuron?
An upper motor neuron lesion.
Right. Okay. So, again, we kind of got this mix of things.
Yeah.
Okay, so they did an MRI on CRN6 as well.
Yeah. Brain and cervical spine MRIs were performed.
Okay.
And the cervical spine MRI showed some chronic changes in the intervertebral disc at C6-7. But there was no significant compression of the spinal cord.
Okay.
The brain MRI, it revealed ventriculomegaly, similar in distribution to CRN4, but it was even more severe.
Okay.
There were no focal T2 signal changes observed in this case. And unlike CRN4, a CSF analysis wasn't
performed.
Yeah, it wasn't performed.
Okay, so ventriculomegaly in both.
Yes.
But other MRI findings that differed a little bit.
Okay. Anything else?
So interesting on the imaging.
That stood out was that both cases showed a diffuse loss of cortical white matter volume.
Okay.
Which was more prominent in the temporal lobes.
Got it. Yeah.
Okay.
So that finding could be significant.
Okay.
But we'll have to explore it.
We'll dig in.
Yeah, we'll dig in.
Okay. So now we've talked about the neurological stuff, right?
Yeah.
Now let's talk about their vision. So both dogs exhibited signs of visual impairment around the age of 4. What did they find on their ophthalmological exam?
So both dogs exhibited thinning of the retinal vessels.
Okay.
And increased reflectivity of the tapetum. So that's the reflective layer at the back of the eye. And based on those findings, they were both diagnosed with an advanced stage of progressive retinal atrophy, or
PRA.
A PRA.
Okay. So progressive deterioration of the retina.
Exactly.
Okay. And this really kind of, solidifies the oculoneurological nature of this whole syndrome.
Precisely. Yeah. These combined clinical findings, they're very complex, but they're crucial for characterizing this novel syndrome.
Yeah. Okay. So before we move on to the genetic stuff, we talked about the parents.
Yeah.
And we talked about the grandmother.
Yeah.
But there were some siblings, right?
Yes. There were 8 pups total in the litter. However, only 4 of the siblings, all males, were available for the study.
Okay.
Yeah. And unfortunately, blood samples from the parents were unavailable.
Oh, go.
But we do know
That they were
unaffected.
unaffected. Okay. So
Yeah.
limited
Sample size right from the start.
Yeah.
Yeah.
Okay. So, but you mentioned
Yes.
that the grandmother, so the parents shared the same mother.
Exactly. That shared grandmother is a really key piece of the puzzle.
Oh, yeah.
Because that pedigree information, despite its limitations, strongly
Points toward autosomal recessive.
Yeah, it points to that autosomal recessive mode of inheritance.
Meaning both parents.
Carriers.
Yeah. Okay. And did they consider other inheritance patterns?
Yeah, we considered autosomal dominant and X-linked recessive inheritance.
Okay.
But both seemed less likely.
Okay.
Given the unaffected parents and the fact that
both were male.
Both of those affected pups were male.
Right.
So
now I have one other thing here. So the owners had submitted DNA from both dogs.
Yes.
To look for inherited retinal diseases in dogs.
Right.
And those tests didn't flag anything.
They didn't.
So that suggests that we might be dealing with something new.
Yeah, a novel mutation.
Wow, okay. So how did they go about finding this?
They started with homozygosity mapping, looking for these large regions of homozygosity, which are areas where the affected dogs shared identical stretches of DNA.
Got it.
And these were absent in the unaffected dogs.
Okay.
And this pointed them towards 7 candidate regions on different
chromosomes.
Yeah, chromosomes.
Okay.
CFA4, 5, 6, 10, 20, 27, and 34.
Wow. Okay. So they kind of had their list of
suspects. Yeah, they had their list of possible hiding spots, for the mutation.
Okay.
So what came next? Whole genome sequencing.
Okay.
Yeah. So they sequenced the entire genomes of both affected dogs and compared them to a reference genome.
Got it.
And then to make sure, to ensure accuracy, they used a reference that included data from over 1,200 dogs, including 6 Cirnecos.
Okay. Yeah. So it was a big data.
A big data set. Yeah. They cross-referenced their findings with another database, the DBMVC, which is a canine variant database. Okay. This rigorous filtering process eliminated false positives and narrowed down the suspects.
So really weeding.
Yeah. Weeding out all the unlikely candidates.
The ones that were
exactly
probably not it.
Yeah, not it. Yeah. So out of hundreds of initial variants, only one coding variant remained that was present in both dogs, both affected dogs, and absent in all the control dogs.
Oh.
So
from both
their internal database and the publicly available ones.
Okay.
And that was drumroll, please.
I'm on the edge of my seat.
A single base pair deletion in the AMPD2 gene.
Right.
Located on chromosome 6. Okay. Within that large...
That was on that list.
Candidate
of
region that we talked about earlier.
Yeah. Okay. Yeah. So
then they designed... they designed a...
Suspect, right.
Yeah. They designed a specific PCR test to check
for
this deletion.
This deletion in all the Cirneco.
In all the available Cirneco.
The DNA sample.
Yeah, DNA samples.
Okay. And what did that show?
So it showed a perfect segregation pattern.
Okay.
Both affected dogs had the deletion in a homozygous state.
Okay.
Meaning they had
2 copies.
2 copies of the mutated gene.
Right.
And then none of the unaffected siblings had 2 copies.
Okay.
But one of them, along with the grandmother, were carriers.
Yeah. Meaning they had
1 copy of the
one copy of the mutation and one of the normal.
And 1 copy of the normal gene.
Got it.
Yeah.
Okay, so this further solidifies the link between
solidifies
the deletion and the syndrome.
Right.
Okay, so now the question is how does this
tiny deletion in the AMPD2 gene cause such significant problems?
Huge problems, in both the eyes and the nervous system.
Yeah.
That is the burning question. That's the crux of the matter, right? To solve this, we have to, we really have to dig into the role of this AMPD2 gene and how it's connected to energy production within cells. So remember this gene. it gives the instructions
Yeah.
to create this enzyme called adenosine monophosphate deaminase 2,
Okay.
which is a key player in this purine nucleotide metabolic pathway.
Right. And we talked about that
Yes.
a little bit earlier.
We did.
But maybe just for those who don't remember or aren't familiar.
Yeah, sure.
Can we break down that pathway a little bit more?
Absolutely. So purine nucleotides are essential. They're like the building blocks of DNA and RNA, right?
Right.
But they also have this really critical role in energy production.
Yeah.
They are essential for making ATP and GTP, which are the primary energy sources for cells.
Right, the energy currency.
Exactly. Yeah, the energy currency. So this pathway, it starts with this molecule called ribose 5-phosphate.
Okay.
And it involves a series of steps that ultimately lead to the production of ATP and GTP.
Okay.
Yeah.
So it's kind of like the cell's power grid, right?
Yeah, exactly.
It's very intricate. And AMPD2
Yes.
has a very specific role in this.
It does. It catalyzes the conversion of a molecule called AMP to IMP
Okay.
within this pathway.
Okay. And what happens when that is disrupted?
So it's like,
By the deletion.
yeah, by the deletion. It's like you're creating a bottleneck
Oh, okay.
in the energy production line. And this disruption can impact the balance of energy production, particularly in these energy-hungry tissues. Like,
Brain retina.
yeah, the brain, the retina.
Right.
Exactly.
And this could explain the symptoms that we're seeing.
It could explain the neurological symptoms
Right.
and the retinal degeneration we're seeing in these dogs. And it's also worth noting that AMPD2 mutations
Yeah.
have been implicated in other conditions, like in mice, for example,
Okay.
hypercholesterolemia and proteinuria.
Okay, so things that you wouldn't necessarily think
Right.
would be related.
Yeah, exactly. So this suggests that the gene might have a broader impact on cellular function than we initially thought.
Interesting.
Yeah.
So, okay, so now we've talked about, we talked about the link to pontocerebellar hypoplasia in humans, PCH.
Yeah.
So can you talk a little bit more about that? Like how similar and how different what we're seeing in the dogs to what we know.
Definitely some intriguing parallels. But there are some key differences. One of the key differences is the presence of retinal degeneration in the dogs.
Right, which is not typically seen.
Not typically.
Human cases.
Reported in human PCH cases.
Right, okay.
Yeah, so the canine syndrome seems to have a broader range of effects.
Yeah, broader spectrum.
Yeah. Another important difference is in the specific MRI findings. So human PCH cases, they typically exhibit abnormalities in the cerebellum and the pons.
Okay.
And these are areas in the brainstem that are really important for, motor control and coordination.
Right. And we're not really seeing that in the dogs.
No, we didn't.
Yeah. See, those classic abnormalities, the MRI findings in the dogs, they mainly showed diffuse white matter loss and ventriculomegaly.
Okay.
And then one of the dogs did have some additional white matter changes and a caudate signal abnormality. But these weren't the classic hallmarks of PCH.
Okay, so it's kind of similar, but not the same.
It highlights the complexities that we can encounter when we're trying to translate findings from animal models to human diseases.
Yeah, for sure. Okay, so why does this matter? Like, this is a very specific breed of dog. It's a rare breed. Why does this matter?
That's an important question. I think this case really underscores the value of studying canine models to get insights into human disease. So,
because dogs and humans.
Yeah, dogs and humans share
a lot of genetic similarity.
Yeah, a lot of genetic similarity.
And dogs, particularly purebred dogs, are susceptible to many of the same
diseases.
Yeah, the same diseases that affect humans. So
it's kind of like having
it's like having the naturally occurring laboratory.
Yeah, a naturally occurring laboratory.
Yeah.
That can help us unravel.
Right, that can help us unravel these mysteries of these
complex diseases. Yeah. Okay, so in this case, this discovery, what is it like? What does it tell us?
I think it expands our understanding of the phenotypic spectrum associated with AMPD2.
That there were probably more problems?
Yeah, it's showing us that mutations in this gene can potentially cause
than we thought.
A broader range of problems than we previously recognized. Exactly.
Right. Okay.
And this case also highlights the importance of considering AMPD2 as a potential candidate when we're evaluating patients, both human and canine, with unexplained neurological symptoms.
Okay.
Or retinal degeneration.
Yeah, so it's like
another tool.
Yeah, another tool in our diagnostic toolbox.
Yeah.
Okay, so we talked earlier about the small sample size.
Yes.
Right. Only 2 affected dogs.
Right.
In this study.
Right.
So how does that affect the reliability of these findings?
That is a valid concern, and while the genetic evidence linking the AMPD2 deletion to this novel syndrome is compelling.
Okay.
Additional studies with larger sample sizes are necessary to confirm these findings and fully characterize these syndromes' phenotypic spectrum.
Yeah, you need more data.
We need more data
to be sure
to solidify our understanding of the prevalence, the clinical presentation, and the potential variability of this syndrome.
Okay, so this is like the first step.
Yeah, it's a first step.
But there needs to be more research.
More research is needed.
To build on it.
Yeah, to build upon these initial findings.
Okay, so how do we do that? Like, how do we get more research?
This is where collaboration and communication within the scientific community are so crucial.
Okay.
So veterinarians, breeders, and researchers, they all need to work together to identify and report any potential cases of the syndrome. The more information.
Yeah, the more information
we could gather.
Yeah, the more data.
The better equipped we will be to understand, to diagnose, and potentially
to treat
this condition in both dogs and humans.
Okay, so it's a team effort.
Yeah, definitely a team effort.
Okay, so let's talk about the breed itself, Cirneco dell'Etna. So this is a rare breed, right?
Yeah.
Yeah, very rare. Okay.
And this rarity raises some interesting questions, like where did this AMPD2 deletion come from? Is it new?
Okay.
Is it limited to a specific family line?
Right.
Or is it more widespread?
Right. Is this...
Yeah, we need to figure that out.
Yeah, we need to figure that out.
Okay.
Okay, so they looked at the genetic history.
They did.
Of the breed.
So to investigate this, they examined the population structure of the Cirneco dell'Etna using genetic data from different populations in both Europe and North America.
Got it.
And what they found is that despite the breed's rarity and the geographical separation, the Cirneco dell'Etna populations in Europe and North America, they're very similar.
Oh, wow.
Genetically.
Okay, so...
Yeah, so even
even though they're far apart, they still have similar...
A relatively closed gene pool.
Yeah, okay.
And
that's interesting.
Yeah,
okay. And
this makes it more likely that the AMPD2 deletion originated from a single common ancestor. Potentially a founder mutation that has been passed down through generations. Yeah, like, tracing the branches of a family tree back to a single root.
And that can tell us a lot about how...
Yeah, it can. It can tell us about... how it spread within the breed.
Yeah, okay.
And then another interesting finding was that they discovered that the AMPD2 deletion was present in dogs of European origin. Okay. Including one that was closely related to the affected dogs in our study. So
it's not just
in North America.
Yeah, a North American phenomenon. It has European roots.
Okay.
And this really underscores
the need for international collaboration to monitor the prevalence of this mutation.
Okay.
And develop breeding strategies to minimize its spread.
Yeah. Okay, so now that we know about this mutation.
Right. Yeah.
What do we do about it?
So the first step, I think, is raising awareness. Breeders, owners, veterinarians, they all need to be informed about this newly discovered syndrome and the potential impact of the AMPD2 deletion.
Yeah, so education is really...
Yeah, education is key.
Important.
Yeah. And then genetic testing is also crucial. So all breeding dogs should be tested for the AMPD2 deletion
to figure out
if they're carriers.
Carriers.
Yeah. This information
it allows
breeders
to make
informed decisions.
Informed decisions, exactly.
Right.
To minimize the risk
of producing affected puppies.
Yeah. So responsible breeding practices are really important.
Responsible breeding practices are paramount. But
as we talked about, it is a rare breed.
Rare breed. So we also need to consider the impact of breeding decisions on the overall genetic diversity of the population.
Yeah, you don't want to limit the gene pool too much.
Eliminating all the carriers from the breeding pool could lead to other genetic problems down the line. Yeah.
So it's kind of a balancing act.
It's definitely a balancing act. You know, it's about finding a sustainable solution that protects the well-being of individual dogs while also preserving the genetic health of the breed as a whole.
Right.
So open communication and collaboration between breeders, veterinarians, and geneticists are essential.
For sure.
To
you have to work together.
Yeah, to achieve this goal.
Yeah. Okay, so, is there anything, like, can we treat this?
So that's the million-dollar question.
Yeah, that's a million-dollar question.
Currently, there are no specific therapies for AMPD2 related conditions.
Okay.
However, this discovery really paves the way for
for future research.
Yeah, for research and development of new treatment strategies.
Okay, so this is
it's a starting point.
Yeah, it's a starting point. One promising avenue is gene therapy.
Okay.
Which could potentially replace
or
repair the faulty.
That sounds very
it might sound like science fiction, but gene therapy is it's a rapidly evolving field with immense potential for treating a variety of genetic.
It's amazing to think that we could potentially correct
these genetic these errors.
Yeah, at their source.
Wow. Okay. So
and then is there anything else, like other areas?
Yeah. Another area of research focuses on understanding the downstream effects of AMPD2 mutations to identify potential drug targets. So, for example, we might be able to develop drugs that boost energy production in affected cells or protect cells from damage caused by energy depletion. These therapeutic avenues, they're still early days. Yeah, but this discovery in the Cirneco dell'Etna provides a valuable model for testing and developing these potential.
So this breed of dog is actually
these rare
helping us to
advance
medical research.
Exactly.
Oh,
it really underscores the interconnectedness of animal
human health. So
okay, so before we
go on
to the
molecular
nitty-gritty details, let's talk about some of the limitations of this study.
Okay, so what are some of the things that we need to design?
We've already talked about the limitations of the small sample size, right?
Right.
And it's also important to note that this study focused on a very
specific breed
of dog.
Right.
And while the AMPD2 gene is highly conserved across species, subtle differences in its function or how mutations manifest in different breeds or in humans are possible.
So we have to be careful about extrapolating.
Yeah, we have to be cautious about extrapolating these findings too broadly.
Yeah. Okay.
And further research is needed to determine
the prevalence of AMPD2 mutations in other breeds and in humans.
Okay.
And to really understand the full range of potential effects.
Yeah, so we need to learn more.
We need to learn more.
Yeah, okay. Anything else? Any other limitations that we need to be aware of?
The limitation is that the researchers didn't have access to tissue samples from the affected dogs.
So they couldn't look at the cells.
Right. So they couldn't directly examine under the microscope.
Yeah, the cells and tissues.
Okay.
To see those microscopic changes caused by the AMPD2 mutation.
Okay. So, but why not? Why didn't they have those?
Well, having tissue samples would have been ideal.
Mm-hmm.
But
Yeah.
obtaining those samples
Mm-hmm.
from living animals
Yeah.
presents ethical
Yeah, of course.
or logistical challenges.
Right. So in this case, the researchers prioritized the welfare of the dogs.
Right. You don't want to put the dogs through any
Because that's
unnecessary procedure.
Exactly.
Okay.
And then lastly,
Yeah.
it's important to remember that
Yeah.
while they identified a single base pair deletion
Yeah.
in the AMPD2 gene,
Yeah.
they didn't definitively prove
Yeah.
that this deletion
Meaning there could be
will
other
cause
the syndrome. There could be other
Got it.
contributing factors.
Yeah.
Either, genetic or environmental.
Yeah. Okay.
However,
Uh-huh.
the genetic evidence, the perfect segregation pattern,
Right.
and the fact that that this deletion has been linked to similar problems
Okay.
in humans really strongly
It's cool
supports
to
its role
Okay.
in this.
Got it.
Yeah.
Okay. So more research is always a good thing, but
All right. Further research.
it's always warranted. But
A starting point.
this discovery provides a solid foundation.
Okay. So now let's
So now let's
get into the molecular details.
let's take a closer look.
Okay.
All right. So remember, we're looking at a single base pair deletion in exon 15 of the AMPD2 gene. And this seems small, but it can have a big impact on the entire protein structure.
Right. We talked about
That function.
mutation. How does this canine AMPD2 compare to the human version?
Yeah. Are there a lot of
Are there
a lot of similarities? There are. The canine and human AMPD2 proteins are remarkably similar. They share 97.34% identity.
Wow, that's a lot.
It is.
Yeah.
It's it really highlights the evolutionary importance of this enzyme and its conservation across species.
So what we learn in dogs
Yeah, what we
can help us to understand.
learn about AMPD2 in dogs can likely be extrapolated to understand its role in humans.
That's fascinating. Okay.
Yeah.
So to understand this specific deletion,
Yes.
you compared the predicted protein sequence of the canine AMPC2
Yes.
to other mammals.
Yes, to those of other mammals.
Yeah. So what did that show? What did that comparison?
So it showed
Those.
that the region affected by the dilution
Okay.
is highly conserved across these, diverse
Okay.
species,
Oh.
which again supports the functional importance of this particular region
Region of the protein.
within the protein. Interestingly, the canine AMPD2 protein is only one amino acid,
Okay.
longer than its human counterpart.
Wow. So just one amino acid difference
One amino acid.
out of, I'm assuming, many.
Yeah.
Okay.
It's a tiny difference considering the overall size of the protein.
Yeah, that's amazing.
It is,
Yeah.
but it highlights the subtle ways in which evolution has shaped these proteins across different species.
Okay. So so we've talked about the gene.
Right.
We've talked about the
protein.
protein. We've talked about what happens when things go wrong.
Right.
So where does this variant fit in? Like the big picture of the AMPD2 gene?
Yeah. So the deletion we've been discussing, it falls within what's called the AMPD domain of the AMPD2 protein.
Okay.
And this domain is really crucial for the enzyme activity. It's essentially like the heart of the enzyme's machinery.
Yeah. So it's not just like
This deletion is like
a problem. It's like a
direct hit.
direct hit to the most important
Functional region of the protein.
of the protein.
Yeah. Okay.
And if we compare this deletion to other known AMPD2 mutations that have been associated with neurological diseases in humans, we see
a pattern.
Yeah. Most of those mutations also cluster within this same
So
AMPD2
disruptions in this domain equal
So there's
neurological problems.
there's a clear connection between disruptions in this specific domain and neurological problems.
Got it.
Yeah. But to really understand the implications of this this deletion, we need to think about the bigger picture, right, of the AMPD enzyme family. So remember, there are 3 AMPD manases in mammals.
Okay.
AMPD1.
Okay.
2 and 3.
So a family of enzymes.
Family of...
Enzymes, each with its own role.
And so do they all kind of do the same thing?
So they're all involved in the conversion of AMP to IMP, which, as you recall, is a key step in the purine nucleotide metabolic pathway. However, each enzyme exhibits a distinct tissue expression pattern.
Okay, so they're not all...
So AMPD1
found everywhere.
is mainly expressed in skeletal muscle, while AMPD2 has a wider distribution. It's found in the brain and the retina.
Okay.
So that
could explain why we're seeing...
specific
you could explain why
we're seeing
neurological...
Yeah, both neurological...
and like ophthalmological issues in these dogs.
Yeah, okay.
And here's where it gets really interesting.
Yeah.
When researchers analyzed RNA sequencing data from both canine and human...
retinal and brain tissue, they found that AMPD2 and to a lesser extent AMPD3 were expressed.
But not AMPD1.
But AMPD1 was not detected in these tissues, which suggests that it might not play a significant role in the brain and the retina.
Okay. So it seems like AMPD2 is the main one.
It seems like AMPD2 is the dominant player in these tissues. Okay. With AMPD3 potentially providing some backup.
So maybe that explains why the dogs aren't as sick as the humans.
This raises the question of whether, this potential redundancy between AMPD2 and 3 might explain why the symptoms in the Cirneco dogs aren't as severe as those, observed in human PCH cases where AMPD2 is often completely absent or severely dysfunctional.
Maybe AMPD3 is helping out.
AMPD3 is partially compensating for the loss of AMPD2 function in the dogs.
Wow. Okay. That's really interesting.
Yeah. It highlights the interplay between different genes, different enzymes, and the complex mechanisms that cells use to maintain homeostasis. It's not always a simple one-to-one relationship between a gene and a disease.
Yeah, for sure.
Now, to add another layer of complexity, it's important to remember that the tissue expression patterns of these enzymes can vary between species. So, for example, studies in mice have shown that while AMPD2 is expressed in the brain, AMPD3 is the dominant player.
Okay. So different species.
This underscores the importance of using appropriate animal models.
Okay. Not all models.
Right. All model are created equal. Exactly.
So, despite all of this, all these complexities.
Despite these complexities and the inherent limitations of any single study, the researchers believe that they have identified a strong candidate for the causative mutation of this novel
ocular neurological
syndrome in the Cirneco dell'Etna.
Right.
And this discovery opens up, a whole new avenue of research into AMPD2 and its role in both canine and human health. It's a reminder that every piece of the puzzle, no matter how small or seemingly insignificant, can contribute to a bigger picture.
Yeah. And this discovery could help,
But this discovery has the potential to help.
Dogs, but people as well.
Not only Cirneco dell'Etna dogs, but also humans suffering from similar neurological disorders.
Okay.
It really exemplifies the power of comparative medicine and the valuable
insights we can gain
from studying animal models. Yeah. It's a testament to the interconnectedness of all living things and the importance of collaboration
between researchers across different disciplines.
Okay. So what are the practical implications of all of this?
What can Cirneco dell'Etna breeders do?
Yeah.
So genetic testing is really important. All breeding dogs should undergo testing for the AMPD2 deletion to figure out
to determine
their carrier.
Yes.
Yeah. This information can help breeders make informed decisions about. Right, about pairings.
Pairings
To minimize the risk.
to risk of having affected puppies. So responsible breeding.
So responsible breeding practices are essential.
They are so important.
Absolutely. However,
Right, but we don't want to.
But a rarity of the breed.
Yeah,
it's also crucial to find that balance between managing this.
Yeah, between addressing this mutation, but then also maintaining
genetic diversity. So eliminating all from the breeding pool.
Balancing act
could have detrimental consequences for the long-term health of the breed.
Okay.
It's
they have to
work together.
Careful consideration and ongoing
figure it out.
Dialogue between breeders and geneticists.
Yeah.
Okay.
And education is important too.
Yeah, education is just as important as genetic testing.
Okay.
Breeders, owners, and veterinarians, they need to be knowledgeable about this newly discovered syndrome, the potential implications of the AMPD2 deletion, and the importance of genetic testing.
Yeah, so the more you know, the better.
So empowering people with knowledge is really crucial for making informed decisions.
Yeah, for sure. Okay, so this has been a really fascinating deep dive.
This case is a valuable
and I'm sure
reminder to all dog breeders, regardless.
There's a lot more to learn
about the significance of genetic testing and responsible breeding.
Yeah, there's always more to learn.
Practices.
So it's a call to action.
To prioritize the health and well-being of our canine companions. Companions.
Yeah, be responsible.
Be responsible.
Yeah.
Stewards of the breeds we love and cherish.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.